This is the Osmosis and Diffusion Lab. The purpose of this activity is to determine how hypertonic, hypotonic, and isotonic environments affect the osmotic activity of cells. To begin, we are going to place 3 pieces of dialysis tubing into distilled water to soak while we set up our beakers. Each one of these is a piece of dialysis tubing. So I'll put it into our distilled water bath. 1, 2, and 3. In beaker number 1, we are going to put 75 milliliters of distilled water. Beaker number 3 is going to get 75 milliliters of distilled water along with 2 full droppers of iodine. We're skipping beaker number 2 for right now. So 75 milliliters of distilled water plus 2 full droppers of iodine. That one was not very full. Let's give it another. There we go. Beaker number 2 is going to get 75 milliliters of glucose solution, and this is 15% glucose. This goes into beaker number 2. Now that our beakers are set up, we can assemble our dialysis tubes. To assemble our dialysis tube, we will fold, so twist, fold, and then twist the ends of the dialysis tubing, and then tie a string around the end. So we got our string, we're going to tie around the end to keep our dialysis tubing closed. Right, and I'll double knot that so that way it stays nice and secure. Right, going to do that for our other 2 pieces of dialysis tubing. The next step is to fill our dialysis tubes. Dialysis tube number 1 will be filled about halfway with 15% glucose solution. So we have our glucose solution. Let's take one of our dialysis tubes. You rub the tube between your thumb and index finger a few times until it starts to open. You may need to use the glass stir rod to poke it open a little bit more. Then once you have it open, add in about halfway full with the glucose solution. So see how there is about half full of the tube with the glucose solution. Then you want to squeeze out all of the remaining air, and you will twist, fold, and twist to close off the opposite end of the dialysis tube. Once you have both ends of the dialysis tube secured, you can lay it down on the S1 marked area of your paper towel. For tube number 2, you will again add about halfway full with the 15% glucose solution. So again, rub the end between your thumb and index finger until it begins to open. Take the glass stir rod to poke it open all the way. And be gentle, it's easy to break these. And then again, add in about half full with the 15% glucose solution. So it's about half full there. So you squeeze out any air, and then again, twist, fold, and twist again to close off the end of the dialysis tube. So tube number 2 is filled about halfway with the glucose solution, and I will put that on the paper towel marked S2. Tube number 3 is going to get some starch solution. So our starch solution will go into dialysis tube number 3. So again, rub between your thumb and index finger to open, so that way the end begins to open. And I realize it's hard to see in the video, but it does open, so that way you can put the stir rod in to get it open all the way. You'll then add in, again, about half full with the starch solution. So fill it about halfway. So it's about half full with the starch solution. You'll squeeze out any air, and then again, twist the top, fold the top over, and then twist again, and secure with the string. So we got our string tied around the end, double knot to secure the string. Now this should go in dialysis tube 3 section there. Now we need to begin recording our data. We will record the initial mass of sac number 1. We'll start by blotting it off a little bit, so don't want any extra water on the outside. Zero the scale, and then place our entire dialysis tube and string on the scale. The initial mass for dialysis tube number 1 is 7.12 grams. We do the same thing for dialysis tube number 2. We blot it dry, zero the scale, and then place the entire string and dialysis tube on the scale. The initial mass for dialysis tube number 2 is 3.82 grams. We finally do the same thing for dialysis tube number 3. The mass for dialysis tube number 3 is 5.97 grams. Tried to change on me there. The next step is to put our dialysis tubes into the solutions in each of the beakers. The dialysis tube number 1 will go into beaker number 1. When we place our dialysis tubes in the beaker, we want it to be U-shaped. So we want to keep the ends of the dialysis tube that we tied outside of the water. In order to do this, we will take a section of tape. So you need a piece of tape. In order to tape the strings or the ends of that dialysis tube up and out of the liquid. So first sack number 1 goes into beaker number 1. And then I will tape, oops. This works a lot easier when you have 2 people. Tape it so that way the ends stay up and out of the liquid. So just like that. If you take a closer look, you can see that the ends of the dialysis tube are out of the liquid, but the body of the dialysis tube is hanging inside of the liquid. We do the same thing for number 2 right away because we want all of these started at the same time. Dialysis tube. Now that all 3 of the dialysis tubes are waiting in their beakers or soaking in that environment, we wait for 30 minutes. Now that 30 minutes has elapsed, it is time to record the final mass of each of the dialysis tubes. We will begin with removing the tube from beaker number 1. Remember, the initial color of the solution in the beaker was clear, and the initial color of the solution in the dialysis tube was also clear. When we remove the tube from the liquid, we see that the liquid inside of the tube continues to be clear, and the liquid inside of the beaker continues to be clear. Dry this off on some paper towel. And then we will record the final mass of the dialysis tube from beaker number 1. The final mass from beaker number 1 is 8.06 grams. Remember, the initial mass from dialysis tube number 1 was 7.12 grams. Now from beaker number 2, We have our tube from beaker number 2. You can see that the tube is still a clear liquid. You can see that the liquid inside of the dialysis tube is still clear, and the liquid inside of the beaker is also clear. Let's pat this dry so we're not getting any extra water on there or any extra liquid. And then record the mass of the dialysis tube from beaker number 2. Remember, the initial mass of the dialysis tube was 3.82 grams. Our final mass is 4.12 grams. So it did not change very much. Then we have beaker number 3. If you look at the color of the liquid inside of the dialysis tube from beaker number 3, you can see it's this dark purplish color. The liquid inside of this dialysis tube initially was a white color. At the end, it's more of a purpley dark color. The liquid inside of the beaker was initially a amber, kind of orangish color, and at the end, it remained that amber orange color. Now let's record the mass of the dialysis tube. The initial mass was 5.97 grams. The final mass is 6.04 grams. For the final part of this lab, we are going to test the contents of the dialysis tubes from scenario 1 and scenario 2. We're also going to test the liquid environment in which they were in. The initial environment for beaker number 1 was distilled water. When we do a test using Benedict's solution, we can test to see what the color change would be for any presence of monosaccharides, so of simple sugars. So I will take a test tube, fill it with some distilled water, which is the same stuff that we put into beaker 1 initially. I will then add in some of the Benedict's solution. So add in a little bit so you can get some good blue color going on in there. So initially we have a bluish color in that test tube. So our initial color from beaker number 1 will be shown in the hot water bath here. So let that boil for a few minutes, and then we will record our data. While the initial solution of the beaker is heating up, the initial solution inside of the dialysis tube was glucose. And this is the same solution that was the initial contents of beaker 2 and the initial contents of inside dialysis tube 1 and dialysis tube 2. So I will use one test tube to test what all 3 of those items would have looked like. So add a little bit of glucose into the test tube. So some glucose, add in the Benedict's solution, so we get that nice blue color, and then add it to the hot water bath. Now, as expected, the distilled water solution is still a bright blue color, even after being in the hot water bath for a few minutes. Distilled water will remain this bright blue color no matter how long you leave it in the hot water bath. The glucose solution in that hot water bath is already beginning to change color. When the glucose solution, that pure 15% glucose solution, is tested with the Benedict's solution, it very quickly turns to a bright orange color. So that is a positive test result. So if we know that there's a whole lot of the glucose solution, it turns that bright orange color. Now we want to test, what do the contents of this beaker number 1 have? So the final color of beaker number 1, even though it looks clear, we need to be able to test to see if this solution that's inside of beaker number 1 contains any glucose. So we have liquid from beaker number 1, add in some of the Benedict solution, and then put this into the hot water bath. A separate test tube is used to test the contents of the dialysis tube that was used in beaker number 1. So I'll remove the string from the end and carefully pour the contents of this dialysis tube into the test tube. I will then add in some of the Benedict solution. So adding the Benedict solution. And I'm ready to place this into the hot water bath. Now, the test tube that was full of liquid from the beaker number 1, you can see is already turning into this dark blue or dark orange color. So I'll let that run just a little bit longer before I put our dialysis tube liquid into the hot water bath. So you can clearly see that there is a lot of glucose inside of that beaker number 1. It's turning a dark orangish red color. This is the test tube with the liquid from dialysis tube number 1. Remember, the initial contents of the dialysis tube number 1 was a 15% glucose solution. You can see already that there is a little bit of a color change taking place. So you can see already that it's starting to turn yellow right at the top. And you can see that the reaction is taking place and is nearly complete. It is not a dark orange color, but it is more of a yellow. So the contents of the first dialysis tube is a yellowish color. Our next test tube will test the contents of beaker number 2. So pour some of the liquid from beaker number 2 along with some of the Benedict solution into a test tube. Now this particular beaker initially had a lot of glucose in it. It was filled with a glucose solution. So we would expect this particular test tube to turn a bright orange color. With just being in the hot water bath for a few moments, you can already see the color beginning to change. I will let it run just a few moments longer. And you can see the reaction is complete. It has turned a bright orange color. And that is the contents of beaker number 2 at the end of the experiment. Our final test tube, we will add the contents from dialysis tube number 2. So carefully remove the string and pour the liquid into the test tube. Now there's not a lot of liquid from this particular dialysis tube. We'll then add in some of the Benedict solution so that way we can see our color change. So we added our indicator solution. We start as the bright blue color, add it to our hot water bath, and wait for the color change to occur. Now remember, this one started with the 15% glucose solution. So we would expect this to continue to have that bright orange color. After just a few moments, you can already see the color change beginning, and it's almost reached completion. At this time, the reaction is complete, and it is a bright orange color, the exact same that it was at the beginning of the experiment with the pure 15% glucose solution.